Thanks for coming to my talk on reusable medical devices and the human factors that are associated with the validations and making sure that all the devices can be processed effectively at the healthcare facilities. So reusable devices come in many shapes and forms. They can be anything from an implant screw to a very large colonoscope. All of these things need to be reprocessed appropriately at the healthcare facility. I've got a few pictures here. This shows human tissue that was found in an arthroscopic cannula during an infection outbreak. And so this shows that there are a lot of issues in reprocessing medical devices. Here's another picture. Even though this device was cleaned appropriately, a brush bristle was left in the cannula, or excuse me, in the shaver. Human tissue and other debris can also be left in the medical devices also. This could be either just from the previous procedure, and also it could be from biofilm that's accumulated over many uses of the device. This is a picture of a surgical suction device, and you can see either there is a biofilm or it could even be a rust in the device. So obviously we wouldn't want this device utilized on us or any of our loved ones. NBC had a special on the Today Show, and it was about a 6-minute video showing the challenges associated at healthcare facilities. I'm not going to show that in the interest of time, but you can definitely find that on their archives. It's on the NBC Today website, but it's a very good little blurb about what the challenges for healthcare facilities are and the infections that can occur when devices are not properly cleaned, disinfected, and sterilized. So I have a lot that I want to cover in this very short amount of time because there's a lot going on in this field. It's a very rigorously regulatory process right now with the FDA. We're just still defining what the FDA wants to see from medical device manufacturers and making sure, our end goal here is patient safety. And so we want to make sure that we are validating correctly and that the healthcare facility can replicate those results. So today I'm going to be going over some guidance for the manufacturers, the outcomes of the How Clean is Clean roundtable discussion that I facilitated, some IFU information, device design for human factors, cleaning validation considerations, sterilization validation considerations, and also a few manufacturers' responsibilities and considerations, which you'll see throughout the whole presentation. So the three most important guidance documents right now for reusable medical device validations are the FDA draft guidance. Now this is still in draft form. I know the FDA received comments for this just a few months after they submitted the draft guidance, and they are working at getting a document that is not in draft format, but it still may be a while for that. They've received a lot of comments and they have worked through a lot of them, but there's still a lot that they're working through to make sure that they can get the most updated information and moving forward that it's going to stay that way. AMI-TR 12 is for the reprocessing of medical devices, and that includes disinfection, sterilization, and cleaning processes. And AMI-TR 30 is specifically for cleaning validations for reusable medical devices. There are also a lot of other working groups associated with AMI and this just shows all of the importance and all the information that is going on with reusable medical devices. There is a lot of standards that are just beginning. There are a lot of standards that are being revamped. They're just continually going through the process, and we want to make sure that everything is up to date. We want to make sure it's appropriate for manufacturers, for healthcare facilities, and also that the FDA agrees with everything that are in these documents so that we can get the information through the system as quickly as possible. So some new guidance that's coming forth through AMI are a standard on human factors device reprocessing. We'll have a draft of this document before the April AMI meetings, so we'll start reviewing that draft. Standardized instructions for use standard is also in a draft form. We've reviewed the first draft, and so we will be talking about the comments associated with that document at the April AMI meetings. And the quality systems for device reprocessing standard is just in an outline format at this point. So we are just developing who should be writing which sections of that document and then moving forward with the draft of that document. There's also a lot of other documents that are coming forth. The endoscope reprocessing document also has a lot associated with human factors. The managing sterilization of loaner instrumentation, that's a huge issue for healthcare facilities, and the fact that they don't always get the instructions associated with loaner instrumentation. They just have to bring it into their facility, process it to the best of their abilities, and then move it through the system. So there's always those issues with the loaner instrumentation too that we're working on. It's such an issue that it's been put on hold because we just don't even know how to tackle it at this point. So that's been very interesting. There's also a lot of information with low and intermediate level disinfectants and their uses. We've got a lot of information on high level disinfectants, but the low and intermediate is where we still need a lot of information associated with that for standards. There's also work being done on the ASTM standards in kind of conjunction with the AAMI standards, trying to make sure that we've got a lot of information regarding test soils, cleanability, etc. So those are coming forth too. So I was honored to facilitate a roundtable discussion on how clean is clean. We had attendees there from AAMI staff, FDA staff, medical device manufacturers, testing labs, and also hospital staff. So we had a really great conglomerate of people. And the big question that we tried to answer, and we're still working on that answer, is how clean is clean? So really, what is the maximum amount of residual soil that can remain on a medical device and still be safe for medical use or safe for patient use? So what is cleanability? It depends on a lot of factors. It depends on the device design, the device compatibility with the disinfectants, the sterilization process, etc. The instructions for use need to be very clear, concise, and also the human factors associated. So this comes down to the essential processing personnel, the equipment, the facilities, etc. So there's two ways to define clean and how to make sure devices are clean going through the healthcare facility. First of all, manufacturers need to validate the cleaning process, and this needs to occur at a laboratory under very strict regimens to get exact residual amounts of markers. So cleaning procedures need to be performed as they would in a healthcare facility, and I'll go through this more as my talk goes on, but we need to make sure that we validate these procedures in a very complex design and making sure that the residual markers are very sensitive and specific for those tests. Next, the healthcare facilities need to verify that the cleaning was performed efficiently in the healthcare facility. Now, this has been difficult for healthcare facilities because there aren't very good residual tests associated. I know that a few manufacturers are trying to make tests that are easy for healthcare facilities to test medical devices, but the problem that they've found is when the devices are cleaned and then tested, then they need to go back through the cleaning process, and healthcare facilities don't necessarily have the time to do that. So that's been a challenge for healthcare facilities. So we really would like a test that would be that you could just test a medical device and then keep it going through the process. And so a lot of manufacturers are looking towards that. So a few more outcomes to the roundtable discussion were that one test soil cannot be used for all medical devices. We would love to come to that. ISO 15883 has been investigating this for years, I want to say 8 or 10 years, to really determine what test soils are appropriate for medical devices. But what it really comes down to is how the device is utilized in the healthcare facility, and we need to be testing the device and with a test soil that's appropriate for that device type. Manufacturers also must need to conduct the validation on devices that are in the used state. So this is, if it's a device that's not on the market, we need to cycle the device through many different cycles of maybe cleaning, sterilization, maybe a disinfection step to get the devices into a used state. We all know that brand new devices with no cracks, crevices, and absolute perfect shape probably are going to be the easiest to clean and sterilize. We want to make sure that devices are at least halfway, through their use state so that we can make it appropriate for all levels of the device. We also need to ensure that the healthcare facility can correctly clean the device. So we need to think about the facilities, the personnel, the equipment, etc. And then we need to take into account with the healthcare facility limits and the device itself. So some devices are very complex, and we need to make sure that there are very good steps in how to perform that test, that cleaning step. So I'm going to go over some information on instructions for use and what is appropriate to make sure that we are providing the best information to the healthcare facilities. A lot of these come from the guidance documents, the FDA guidance documents 17665, 17664, and this is to make sure that all the information on an IFU is make sure it's grammatically correct, make sure all the information is legible, that it's presented in like first step to last step. We need to make sure that it's at a 6th grade reading level. A lot of the healthcare personnel in the central processing area do not have more than a high school diploma. So we need to make sure that it's very easy to understand. We also need to use showing in Arabic numbers to show the progress through the cleaning steps, and also use images to show the essential steps. And this also could include videos and so on to really make it easy for healthcare facility people to perform the cleaning procedures. A few words associated with IFUs are just not allowed anymore, and that is a minimum of, if appropriate, if possible. We find this a lot of times. If the device isn't cleaned thoroughly, then use a minimum of 1 minute to clean it. We need to have very specific numbers to make sure that the devices are appropriately cleaned. Another important aspect of cleaning may be disassembly and reassembly of the devices. We need to have step-by-step instructions on the IFUs, and these also could include illustrations, diagrams, videos, and so on. And we need to make sure that this occurs because disassembly may be vital to cleaning the devices. Some more information that needs to be included on the IFU are the detergent dosage, so the exact ounce per gallon of water, the specific temperature that the cleaning should occur, the water quality, the exact time that each step should occur, whether it's brushing, flushing, etc. The brush type and size, if we need any brushes to clean lumens or outside cracks and crevices and so on, we need to have that exact brush type defined, and part numbers included in the IFU if those products are not provided with the device itself. So some kind of new expectations for manufacturers, device manufacturers from the FDA, are that the FDA expects manufacturers to come on-site at healthcare facilities and provide on-site training and education for sterile processing personnel. They find that this not only teaches the central processing staff on how to clean the devices, but it also provides a really good avenue for feedback regarding the device types and how they can be sterilized in healthcare facilities. So FDA is really pushing for healthcare facilities to perform that right now. As part of that, then they can get feedback from the healthcare personnel, sales reps, etc. And then they can also develop education, training, and competency verification. And that's very important to continued verification of people's abilities to properly maintain the cleaning procedures of all medical devices that come through healthcare facilities. One thing that we need to keep in mind while test labs are performing the validation process and when the healthcare facilities are performing the cleaning process itself, that there are very strict PPE that healthcare facilities need to use when they are performing the cleaning. So we have to make sure that the cleaning procedures allow for those heavy-duty gloves and all the facial components and all the masks and so on, and the big gowns and the hot hot rooms that they're cleaning in and so on. So we need to keep into account all of that information also. So in conjunction with designing medical devices, we need to make provisions for cleaning and sterilization, and they should be considered in the very first stages of the medical design process. We can't just make a device that is going to work perfectly in a surgical setting and find that it cannot be cleaned and sterilized properly. That just is a very difficult situation to overcome when we have to make modifications to devices when we want that device to be utilized in the healthcare facility. So some device design considerations that make cleaning difficult are areas that harbor unwanted organisms or organic material. And these include braided and twisted wires, any kind of textured surfaces, which include hinges or springs. Dead-end lumens and channels are definitely difficult to clean. These can be flushed, but if they don't have a way for the water or detergent to escape the other side, it's hard to make sure everything is coming out the one channel. Inaccessible cracks and crevices are definitely a challenge to clean, and also lumens and O-rings. So they're all very difficult to make sure that we have the correct. These are features that make cleaning very difficult. Some materials that make cleaning difficult are silicone and rubber and aluminum-based materials. These are probably more they have more effect on the sterilization process than the cleaning process because of their thermal conductance. So here's an example of a metal medical device, and you can see, so this dark blue line is the heating of the device itself. And so you can see the ramp up was very much lower than the first pulse. And by the second pulse, it almost makes it to where it should be. And then by the third pulse, it's right there. So it took a little bit more time to ramp up than the sterilizer temperatures, but by the third pulse, it was right there. And then the exposure period, it stays exactly where it should be at the temperature required. Here's an example of a silicone device where it takes a lot longer to heat up. Even by the third pulse, it's not making it where it should be at the high sterilization processes during the third pulse. And then by the exposure time, it is just under where it should be. So we're looking at, only 128 degrees for part of that exposure period. So that just shows it takes a lot longer to heat up than a medical device versus a silicone device. So some other complex factors of medical devices that may create opportunity for error would be devices that cannot be disassembled and reassembled very easily. Some that have a ridiculous amount of parts. It's hard to make sure that everything gets put back into its place. And then, if it doesn't, then it'll go into the operating room suite, and the doctor may not have everything that they need to conduct that sterilization process. And this confusion also extends the processing time, which is already still very, healthcare facilities are working really hard to make sure that they process things very quickly and get them back into circulation. And so we can't make that process any longer than at all possible. So we know a lot of complex devices have unavoidable problematic design features. And so we just have to work with those in the validation process to ensure that we can clean and sterilize and disinfect devices properly. So I'm going to go through some cleaning validation considerations. I'm going to be talking a little bit about worst case testing, appropriate test soil, and appropriate residual testing. So really the questions that we need to ask for the validation process, and this is the one that the manufacturers perform when they validate their devices through the very stringent laboratory settings, is what test soil should be used. And I've already spoken about this a little bit. We need to make sure that we need to think about whether the device will come into contact with blood, mucus, cerebral spinal fluid, brain tissue, etc. And we need to design our test soil according to that situation. We also need to use clinically relevant soils. Now this is a huge buzzword for the FDA. We need to make sure that the test soil is appropriate for the clinical setting. And so it needs to have the same contents as the device would come into contact at the healthcare facility. The manufacturers need to really think about this process and justify why the test soil is appropriate. So next, after we've determined the test soil, we determine what the contamination process will be. So we need to determine, does the device get immersed into the body? Does it just get handled by the physician? What process does the device go through to make sure that we are considering a worst case contamination? Part of this process is to think about worst case contamination, but also to make sure that we aren't doing a ridiculous case contamination. Sometimes what our manufacturers are being asked by the FDA is to do even worse than the worst case, and that is making the cleaning process extremely difficult. It's increasing the cleaning parameters, thus affecting the healthcare facility and the healthcare facility's ability to clean devices. So we've got to find a balance between there to find the worst case testing and not the ridiculous case testing. The wet soil, we need to determine how long the device is going to be utilized in the surgical procedure. So we need to decide how long the device will be in contact with the soil. And then also, how long will it be between the surgical procedure and when the device will be cleaned? And what is that time frame, and what will the device be in contact with in between that time frame? So that all needs to be determined up front before the validation occurs. So there are a lot of methods of cleaning. There's a lot of information in performing the IFU. Manual cleaning is mandatory. Automated cleaning is mandatory if the device can go through automated cleaning procedure, and if it does, then that's a mandatory procedure also. There are many different types of cleaning markers associated with whatever the device will come into contact with, but the most important markers that are utilized right now are protein, hemoglobin, and total organic carbon. Those are the ones being accepted by the FDA the majority of the time at this point. So for acceptance criteria for cleaning validations, there's really no established performance criteria. These validations need to be predetermined and justified by the manufacturer. AAMI TR 30 has good benchmark validation points that are very well accepted by the FDA and what most manufacturers use as their acceptance criteria. We just need to make sure that we are thoughtful in the process of determining what that acceptance criteria is and defining that prior to the validation process. Some information that we definitely need to add to the cleaning validation is all accessories required, the techniques used for rinsing, brushing, and flushing, the water quality, concentration of chemicals and detergents, and the exposure time and temperature of each step. So I'm going to quickly go through some information of sterilization validations. There's many types of sterilization that occur at healthcare facilities. The sterilization validation steps are fairly simplistic. This is a part of the validation process that has been, 8 or 10 years ago, we were where we are now with cleaning, and we just didn't know what to do with the validation process of sterilization either. But now it's very well defined, and it's very simplistic to put devices through. So we perform a sterility assurance level validation with half cycle parameters. Then we perform dry time validations with full cycle parameters, and then temperature profiling if it is a case system. We place biological indicators in the most difficult to sterilize areas, and those include lumens and mated surfaces. And for the sterilization validation acceptance criteria, we need to make sure that all validation cycles must be within specifications. All SAL cycles must demonstrate a minimum of a 6 log kill, which then results in a sterility assurance level of 10 to the negative 12. Packaging and products must pass visual inspection, which means no moisture on any part of the device or packaging, and all packaging weight gain may not exceed 3%. And there are specific calculations associated with that. So the recent sterilization guidance from the FDA is that we need to make sure that all sterilizers, accessories, et cetera, that are used in the validation can be used in the healthcare facility, and all have K numbers, so they're all FDA cleared. The parameters for sterilizations must fall into the parameters that are approved in AAMI. There's different standards that have that, and the FDA is not allowing any more extended cycles. So we need to make sure that the parameters for sterilization fall within the recommended parameters and that we are not exceeding those. Here's an example of one of the charts in AAMI ST79 that shows the parameters. The most common is the wrapped instruments, 4 minutes at 132 degrees and drying times at 20 to 30 minutes. This number can be extended slightly. We've seen everything to about 45 minutes get through the FDA, but we need to make sure it stays at that level or lower. So some sterilization considerations is we need to make design so that the material of the devices heats up quickly and remains hot. We need to limit blind holes and dead-ended lumens. We need to make sure all of the parts of the device allow for steam penetration and proper air removal. And we need to make sure that ensure the sterilant can flow through the entire device. And kind of the same for the tray. I mean, the tray provides additional challenge to make sure that we are getting all the sterilant into every crack and crevice, the air out of all the cracks and crevices, and the sterilant into all the cracks and crevices to make sure everything's sterilized appropriately. We also need to make sure that the layout and the tray design is appropriate for sterilization. And this ensures proper sterilant flow, that we don't have any mated surfaces that do not allow for the sterilant to penetrate. We also need to make sure that everything is in the open, unlocked position, and that the mass be distributed through the trays, not only for sterilization, but for easy movement around the healthcare facilities, so that we're not, tipping trays as we move them around. For the dry time testing, we need to think about the fact that materials that stay hot do the best. We need to reduce the mass. We need to not allow areas that may pool water, and if there is any pooling of water, we need to make sure that appropriately drains. And I've covered most of this information, but this is just information associated with ISO 17664 that's necessary for sterilization validations. This includes the temperature of the sterilization agent, the humidity, the exposure time, the pressure, and all the information associated with the validation needs to be put into the IFU to make sure the healthcare facility can follow that directly. So this is the 7 criteria for reprocessing instructions that comes right out of the FDA reprocessing document, but it really reflects on what we need to do to make sure that devices are appropriately cleaned in healthcare facilities. And so we need to make sure that the labeling of the device reflects the intended use. We need to make sure that there is a thorough cleaning process, a thorough microbiocidal process, that the reprocessing steps are technically feasible, and we can do this through going through the healthcare facility to help that process. You need to use all disinfectants and detergents that are legally marketed and have FDA approval. We need to make sure instructions are comprehensive and understandable. So it's very important. All right. If there are any questions, I would love to entertain any questions.